Thank you all so much for joining our Intro to Terra webinar. I'm Elizabeth Kiernan. I'm a science writer for Broad, and in a few moments I'll be introducing my colleague Anton, who will get us started with our presentation.
Thanks very much, Liz. As Liz said, my name is Anton Kowalski. I'm also a science writer for the Broad Institute's data science platform. Today, we have a webinar for you about Terra, which is a platform for biomedical researchers to help them access data, run analyses, and collaborate together. And just to give you a brief overview of today's session, we'll start by introducing the concept of Terra as a platform, which means actually first we'll start by introducing the concept of cloud computing in biomedical sciences. And then I'll hand it back over to Liz, and she'll describe to you how the Terra workspaces work and what types of modes of computation are available. We'll, at that point, actually pause the slide deck, and we will go into the internet browser, into the actual web application. We'll set up our own workspace and launch a runtime environment. And then we're actually going to pause there and come back to the slide deck while the runtime environment is getting spun up, as that takes a little bit of extra time. So while that's happening, we'll just go back to some high-level information about security, access, and billing, and then we'll go back to our web application, and we'll actually do a hands-on demo where we'll get to launch some computational workflows and work from some Jupyter Notebooks. So we'll start by setting up the problem. So the big data problem really comes from the fact that biomedical research requires the generation and storage of increasingly more vast amounts of data. This is because a lot of this work is done by many individual labs as well as by large community-wide projects. And as the data structure grows, many problems involved with the format and interoperability begin to proliferate. So in short, how do you standardize the formats that everybody is working with so that, first of all, everybody is able to read every piece of data, but also so that everyone is able to perform computation in a reproducible way. And the proliferation of the data and the code to do all of this is getting so big that it's becoming very impractical to try to download everything on your own. So another way to look at this problem is to compare the traditional way that biomedical researchers have interacted with data with what we refer to as the cloud paradigm. So in the traditional paradigm, we bring data to the researchers. So for example, let's say you have some very large chunk of data, and I'm very interested in doing some research with that chunk of data. I could come to you and ask for your permission to copy that data, and hopefully you will agree. And then I'll get to have my own copy of the data. This, of course, is inherently inefficient because now we have two exact copies of the exact same data, which shouldn't be necessary. In the cloud paradigm, instead of data copying, we have true data sharing. So we stand up this large piece of data in a central location and then create a system of access that allows researchers to come to that data and do whatever it is that they need to. And so this applies both to the concept of data copying versus data sharing, but also in general to other things such as the requirements of having infrastructure to handle all of this data and all of the computation. In the traditional way, everyone would have to have their own enormous infrastructure to handle all of their needs, whereas in a cloud-based infrastructure, everyone can share in the resources. Another thing that this leads to is the elasticity of computation in the cloud. So in addition to how convenient it is to have a central place where everybody comes for their data and code, This is also a way of getting around the problem of spikiness in data usage and computation. So traditionally, there are peak times when a lot more computation is performed, especially in preparation for big conferences, things like that. And so in the traditional format, everyone had to have their own infrastructure, and everyone's infrastructure had better be able to handle both their low and their high usage times. Whereas in cloud-based computation, we have a lot of computational elasticity, as we refer to it. So you are able to requisition whatever is the correct amount of computational resources for your needs at any given time. So how do we leverage the cloud to solve these problems? These institutions came together and laid out a vision for what they refer to as the data biosphere, which is kind of like a vibrant ecosystem of all of these data repositories and the code to handle them. And everything is designed to be in accordance with standards laid out by the Global Alliance for Genomics and Health. So the authors represented in this article are actually representatives of those institutions that I just mentioned. So they got together and they laid out their vision and penned this article, which I highly recommend everyone to go check out. It's not a peer-reviewed journal article or anything like that. It's a very fast read. You can see the URL where you can find this article right here. And overall, this article lays out the vision for the ecosystem, which contains modular and interoperable components for a diverse data environment. And ultimately, they laid out a vision that corresponds to these four governing principles. The first is that the data biosphere has to be modular, so it has to contain a lot of interoperable components with very clearly defined interfaces, so that they are so that it's easy to tear apart pipelines and put them back together in whatever way you need. Number two, it has to be community driven. So we have a lot of feedback loops with the community. We really strive to make sure that we're answering the questions that the scientific community has at the moment. We try to stay up to date. Number three, everything has to be open in order to enable reusability and extensibility for the scientific community. And finally, of course, everything has to be standards-based. So we make sure that our platform is in accordance with the standards for interoperable interfaces laid out by the the GA for GH. So that gives you an idea of what the data biosphere is supposed to do. And here is a diagram that lays out the data biosphere at a glance. So down here we have the data assets to which we want to link scientists. And at the very top are our users, you guys. And so where does Terra as a web platform fall into this architecture? Well, Terra is the platform that is supposed to link the users to the data assets. And that's what today's webinar is really about, is showing you around the Terra user interface so that you can see how exactly you can be connected to all of these data assets and how you can make the most use of them. So just to give you an idea of what types of data assets Terra gives you access to, here's a list of some of the data sets that you can access through Terra, as well as the institutions that are responsible for those data sets. And that brings us to the concept of Terra itself and the workspace, which is the reproducible digital laboratory in which you can do all of this work. And so with that, I'm going to hand things over back to Liz, and she's going to tell you a little bit about workspaces and what you can do inside of them. And then we'll go ahead and do a little bit of hands-on work.
Great. Thank you, Anton. I'm excited to get a little bit more now into the nuts and bolts of what Terra can do, starting with the workspace. So you can think of the workspace as a computational sandbox that lives within the cloud environment. It's an area where you can manage your data, organize it, you can run pipelines or workflows on your data, and you can use notebooks or Jupyter Notebooks to interact with your data and visualize it. At the heart of the workspace is a data model where you can describe what types of data you have and where they are located. So every time you generate a workspace in Terra, you're going to also generate some workspace storage in the form of a Google bucket. But you can also have your data already existing in a Google bucket storage that's outside of Terra, or you might be pulling data in from a public repository. I just like to emphasize this, that you don't have to re-import your data into the workspace storage. You can access it from Terra if it's in one of these other repositories, saving you time. When you come into a Terra workspace, you will come to a landing page that we call the dashboard, and it looks very similar to the picture on this slide. What I want to point out is you'll notice a few tabs at the top here. Under the dashboard where we are right now, this is where you can identify what types of data are in your workspace and what types of analyses you can run on those data. In your data tab, this is where you can actually go in and upload tables containing the metadata, so telling what type of data you have and where it's located. Under the Notebooks tab, this is where you can actually launch a Jupyter Notebook to do analyses. In the Workflows tab, you can select a workflow, an existing workflow. And in the Job History tab, you can look at workflow provenance, how a workflow is progressing, or any errors that may come up. You'll hear us talk a lot today about Jupyter Notebooks, and you're going to get to actually see a Jupyter Notebook in action in the live portion of this workshop, of this webinar. But for those who don't know, a notebook is essentially a digital version of a lab notebook. It's an area where you can describe different types of analyses using markdown fields like this one shown here at the top. And you can actually use code blocks to run code and interact with your data in real time. One of the great aspects of Terra is that we have an entire section dedicated to showcasing featured workspaces. These are workspaces that come preloaded with data and analyses, everything that you would need to do to get hands-on right away with some of these different bioinformatic tools. We're going to be exploring these workspaces today for the hands-on portion, but I encourage you to come back to this section afterwards and take a look for yourself, because again, it's a great way to just get started right away with understanding Terra. It's very easy to collaborate and share using Terra workspaces. By default, the workspaces are private, and you can give individuals or collaborators other forms of access permissions. Three main forms of permissions that you can give your colleagues are reader, writer, or owner. Readers can go in and look at a workspace, see what kind of data you have and what types of analyses you're doing, but they can't actually edit anything within the workspace, so they can't change your code or anything. A writer will have edit permissions, but they can't share the workspace with other colleagues. And an owner can both edit and share a workspace. These are the three main permissions, but I want to emphasize that you can use different variations of these to customize permissions as you need them for your purposes. All right, there are several modes of computation that work within Terra. So we've sort of touched on this, but you can run a large virtual machine within Terra. So you can launch a Jupyter Notebook and run R or Python on your data. You can also run shell scripts on a terminal in a virtual machine. You can perform large-scale analytics. So you can load tabular data like that found in BigQuery. For those who don't know, BigQuery is Google's database, public database, that holds tabular data like phenotypes and medical records. In our hands-on demo today, we're going to be exploring some BigQuery data using a Jupyter Notebook. Lastly, you can perform batch computing in Terra, so pipelining. And a lot of times these pipelines can take a while to run, but one nice aspect is that you can close up Terra and walk away and your pipeline will continue to run. Your workflow will continue to run. I do want to go a little bit more into this idea of batch computing and running pipelines, because for many of you who actually do this, you know that it's very important for these processes to be automated and reproducible. I'm showing you an example here on this page from the GATK for best practices pipeline. The goal here is not for you to read all these steps within this pipeline, but just to recognize that there are many steps and that each of these is its own bioinformatic tool. If you had to go in and manually run each of these steps on your data, not only would it take a large amount of your time, it would also introduce a lot of room for human error. So we want to make this process as automated as possible, and that will also help us reduce time re-implementing the wheel if we need to make a change to any one of these different tools that are used. Terra uses a couple of pipelining products that assist with this automated processing. So I'm going to take a few minutes to go into those products. The first one is Whittle, a workflow description language. The second product is Cromwell, an execution engine. And the third one is Docker containers. So we're going to go a little bit more into each of these products. Whittle was created, the workflow description language was created to be a human-friendly scripting language. It starts with a workflow, and that workflow can have inputs shown in yellow, and it will execute various tasks. And it does this by using this call function here. So you can see it says call task A. If we look more closely at these tasks, you can see that they're very similar to functions. So each task also has inputs shown in yellow and outputs shown down here at the bottom. And you can run commands on these tasks, which will be what type of transformation you want to put the inputs through. For each task, you can set a runtime, which tells the command exactly what is necessary in order for that command to execute successfully. And in this case, it's calling a Docker. When you run a workflow, you see that some of these inputs can be called from the tasks or the workflow themselves, as is happening in task A here. But you can also call inputs from the output of previous tasks. So if you look at this task B, you can see that the input for this is a task A out. So this is an example of a linearly chained script, but I do want to emphasize that you can run these tasks in parallel as well. So it can match whatever pipelining needs you might have. What's great about Whittle is that it runs on Cromwell, which is a portable execution engine. This is important because portable meaning it can run both from your local computer, but also within the cloud environment. So when we are trying to think about making scripts more reproducible, the fact that it can run from any location is very important. Cromwell does recognize Whittle, but it also recognizes common workflow language if you have any interest in that, and it might recognize other languages in the future. And I just want to point out that if you have any questions about these pipelining products, the links to more information are found at the bottom of the screens here. All right, the last product I want to talk about that Terra uses is Docker containers. For those that don't know, a container is a package that contains software and all of its dependencies. So if you think of a situation in which you've ever tried to install software on more than one computer, maybe you ran into a problem where you didn't have the right form of Java and your software couldn't install the way it's supposed to. This is a big problem when we're talking about analyzing data, because if a software doesn't install appropriately, it could actually, in fact, change the way your data was analyzed compared to a colleague's. So Docker is sort of our solution for this problem because it allows you to share software and its dependencies the exact way that you ran it with a colleague. So this is, again, another feature that Terra uses to help make this pipelining a little bit more automated and reproducible. So just to kind of summarize what this allows us to do, it means that researchers can use Whittle and Cromwell to do some development of scripts and some small scale testing from their local machine. But using Terra, they can also use these same pipelining products to do all major production on the cloud environment. Okay. So let's go ahead and explore now the Terra and get a little intro to some Terra workspaces. So I'm going to switch gears here. All right, and if you're following along, feel free. I'll try to follow along on your own computer. I'll try to go slow so that you all have time. So I want you to go to app.terra.bio. This is the home page for Terra. And you see when you enter this homepage that there are several quick start buttons here at the bottom. If you've already signed up for Terra, you can immediately go to your workspaces using this first button. You can view examples like those featured workspaces I spoke about earlier by clicking View Examples, or you can browse data. But the most important part of this Welcome to Terra homepage is the hamburger button at the top left. So when you click on this button, you'll get a drop down. You'll see right away that you can access your profile. This is where you can change billing and group information. You'll see that you can access your workspaces. If you go into this for the first time, you'll probably find that your workspaces are empty because you have not created a workspace yet, or you have not cloned a workspace from another person. You'll also see this library option. If you click on this dropdown, you'll see three options. Data, this is where you can go and explore public data repositories that are available. Showcase, this is where we have our featured workspaces. And you'll see workflows. This is where you can go and select an existing workflow written in those Whittle scripts that I spoke about just a few slides ago. You'll also find that you can sign up for your Terra free credits here. If you haven't done this already, this is a promotion that we have going on. It's 300 free Terra credits that last 60 days from the time you start up. You can use those free credits to run our hands-on demo today as we're doing this. So if you haven't signed up, please go ahead and do that if you're interested. Lastly, you'll see on this hamburger menu the support drop-down, and this is where you can come to get how-to guides, to request features. We have a community forum where you can post questions, and of course, you can contact us at any point with questions. Anton will show you these features in a little more detail at the end. Okay, so right now I want to go ahead and show you our featured workspaces section because this will be really beneficial for getting hands-on in Terra. So when you go into here, you can see that we're on the showcase and tutorials. But if you look at these other tabs, these are those data, the data tab where you can go in and look at public repository data. And you also see the code and workflows tab here where you can go in and select those Whittle scripts. So I just wanted you to see that you can access that from the top here as well as from the hamburger menu. Inside of our featured workspaces page, you can see two columns of featured workspaces. On the left, we have our GATK4 example workspaces that again come preloaded with data so that you can look at these different components of the GATK4 best practices pipeline. So each one of these is a different tool that's used and you can go in and explore that. On the right-hand side of this showcase are featured workspaces that cover a range of use cases. So anything from DNA methylation preprocessing to single-cell RNA sequencing. I encourage you all to go in and see if you can find a featured workspace that might meet your computation needs, because again, it's a great way to get started. For today, we're actually going to be working on the featured workspace Terra Quick Start. So we're going to click onto this. This is a workspace designed to get you started with Jupyter Notebooks and workflows, and it also shows you how to populate a data table. When you click into this, in the coming soon in the future, you will see app queues that will actually guide you through this particular workspace. This isn't true for all of our workspaces, but it's an exciting feature that we have coming soon to hopefully help get you started faster. For today, I'm going to close out of this. Okay. So now we're in the Terra Quick Start workspace. We know that because we can see the title of this workspace up here at the top. And you'll notice that right next to the title on this top banner, it says read only. That's because I'm not an owner of this workspace. And if I want to go in and actually edit this workspace, I need to make a clone of it so that I can be an owner. So to make a clone, you go to this 3-button icon to the right and click on it and say clone workspace. When you do that, you'll be prompted for some information. So the first thing we need to do is make a unique name. I'm going to call my workspace webinar and my initials LK. But you can feel free to name your workspace any way you want. You need to select a billing project to use this workspace. This is where your Terra free credits will come up if you select it. Mine have already expired, so I'm going to use a different billing project for this. The description is just simply a description of the workspace that you can choose to fill out if you want to change anything about it. Authorization domains are not important for the purpose of this demo, but Anton will go more into what this means in a little bit. So let's go ahead and clone this workspace. Okay. So now that our workspace is cloned, I just want to point out up here at the top banner, now it no longer says read only, so I know that I can go in and edit it. And additionally, if you go down to the middle of the right here, you'll see owners and my name is under there. So if this is done correctly, you should see your name there instead for your cloned workspace. If you want to follow along with these exercises that we're going to show you today, all of the information you need is throughout this dashboard. So you can see we're going to be doing several different types of analyses in notebooks and using workflows. And there are specific instructions and exercises further down. So this is what Anton will walk you through today. If we go to the data tab, you'll see that this is where you can upload different types of tables that contain information about your data. So Terra has sort of divided off 3 sections for different types of tables. This first section that just says tables, this is typically where people will upload their actual sample data. So we can just take a look at a sample one of these. You can see that in this sample table, there's a sample ID. And then over here it says CRAM. These are CRAM files, which are compressed BAM files. You can go in and edit these actually within the table, so you can change the names from within Terra. And if you go into the CRAM file cell, what you'll see is that this actually tells Terra where this file is located. So if we scroll over, this GS indicates that it's in a Google bucket. You can also see that you can modify the attribute types for this data entry. If you will be working with reference data, say if you need to align data to a genome and you need reference FASTA files, you can find human reference data preloaded in Terra. So we have 2 options. There's B37 human and HG38. So if you select either one of these, it will populate a data tab with everything you would need for working with human data. But let's say you're interested in working with mouse data instead. You can upload a data table to the other data section that can contain FASTA files or other for your particular species. In this workspace table, you can see that we have just the Homo sapien HG38 FASTA files present. And if you have any questions more about data tables and populating this, Anton can also answer that a little bit more in detail at the end of this webinar. All right, if you go into the Notebooks tab, you see that for this Quick Start workspace, we have 4 notebooks for you to look at. And this is, Anton will be setting up this R environment notebook in a few minutes, but I just wanted to show you that when you enter these notebooks, you'll see three options: a preview option, which is read only, an edit option, and a playground mode. If you want to work within the actual notebook and make edits, you'll have to start a virtual machine, which is what we're going to do today for this hands-on demo. The playground mode will also launch a virtual machine, but any changes that you make in playground mode are not saved. So that's the difference between edit and playground mode. All right, let me go back to our tabs here. All right, in the workflows tab, again, this is where you can see any WDL scripts that are in your workspace. And so you'll be running these two workflows today in the hands-on demo. And again, the job history tab, this is where you will find any errors or workflow providence or progress. So as you're running these workflows today with Anton, feel free to click over and check your job history as they're running to see more about what that looks like. Okay, I think that is pretty much the end of our digital tour. So now I'm going to hand this over to Anton to get started with our actual demo.
Thank you so much, Liz. Okay, so hopefully everyone has been following with us. Let us know in the chat in the Google Hangout window if you're experiencing any trouble, and we should be able to help you get up to speed with us. In the meantime, what we're going to do is launch our notebook runtime. I'm going to show you that there are a couple of different ways to do that. And also you'll see that there is about a 5 to 10 minute downtime after you initially launch the notebook runtime while your computational resources are actually requisitioned. And so in that 5 to 10 minutes, we'll just go back to the slide deck and talk a little bit more about some high-level things like security and billing. But in the meantime, we'll go ahead and launch our notebook runtime. So as Liz pointed out, this notebook runtime button can be found in any tab. While you're in any tab inside of a given workspace, the notebook runtime is associated with the workspace, but applies only to the work that you do in a Jupyter notebook. So one way to launch a Jupyter Notebook runtime environment is to click on the button itself. It brings out this large panel to give you an idea of what sorts of custom options you have. In fact, I'll go ahead and select the default Python option. We also currently offer a Docker image with Bioconductor pre-installed. This is also where you can go to increase or decrease the computational resources that you are requisitioning. You'll notice that there is a real-time cost estimate down here at the bottom. So in the default mode, a Jupyter Notebook should cost about 19 cents per hour. If I were to click high compute power, it would go up to about $3 per hour. We're going to stick with the default moderate computational power that we need. And actually, rather than launching it from here, I want to show you that the notebook runtime will automatically launch as soon as you try to enter either edit mode or playground mode. So as Liz mentioned, and I'll go into just a little bit more detail, when you first click on a notebook, you see a read-only preview of it. That's, of course, just to show you what notebook you're actually looking at in case you have a lot of different versions, so that you don't have to wait 5 to 10 minutes to spin up a cluster, only to find out that you actually opened the wrong version of a notebook. So you will always be able to see a preview immediately. And if you want to actually edit the notebook, then you'll have to go into the edit mode. We have both an edit mode and a playground mode to help collaborators, basically to help them not overwrite one another. So if you are working in a particular notebook in a particular workspace and you go into the edit mode for that notebook becomes locked so that no one else can make edits in that notebook until you have left the notebook. But if you are working with a lot of people on the same notebook and one person happens to have it locked, but a lot of other people want to be able to go into that notebook and play around in it, they can open that notebook in playground mode. That will still necessitate spinning up a cluster for them to use. However, none of the changes that they do during playground mode will actually be saved. Whereas in edit mode, everything is automatically saved every couple of minutes. So I'm going to go ahead and click on the edit mode. And as you can see, it says, I have a message that says starting notebook runtime. This may take up to 2 minutes. For us, this will take up to 2 minutes because we have already started a runtime environment like this before. For those of you who are doing this for the first time, it could take, the message should say that it would take up to 5 to 10 minutes. In some very rare cases, it can be it can be 15 minutes. But once you have done it the first time, every subsequent time should only take about 2 minutes. And you can see up here an indicator that tells you whether or not your notebook runtime is currently running, whether it's currently starting up. And these also provide you with the buttons to pause the runtime so that you are not needlessly wasting money. So we're just going to wait for that to load up. That's going to continue loading up even if I go to another view of my workspace. So there I just went back to my dashboard. You can see the notebook runtime update is still in progress. So with that, I'm just going to go back to our slide deck and we'll talk a little bit about security, access, and billing in Terra. So of course, we'll start with security, which is of course a huge priority for the entire biomedical research community and for the Broad Institute especially. So to that end, Terra is certified FISMA moderate from the NCI. In addition, we also leverage the Google Cloud Platform's FedRAMP authorization. And so Terra configures all of these resources appropriately so that you can work confidently on the cloud and not be worried about any types of access problems with your sensitive data. So that brings us to access. Access and permission in Terra is controlled by assigning users roles that gain them access to resources. In Terra, there are a few different things that are considered resources. Most importantly, of course, workspaces are considered individual resources, and every individual user has a different type of role assigned to that particular workspace. So over here, I have a screenshot of the panel that you saw on the right hand of the dashboard view. So you can see that in this example workspace, the person who took this screenshot had an access level of reader, and these are the emails of the owners of that workspace in case that reader wants to send them a message to either let them know that there's a problem with the workspace or to request additional access. Other types of resources in Terra are billing projects, which are your actual billing accounts, as well as collections of workflows and groups. These are all types of resources which the users that create them control. Specifically, they control what other users get which type of access to those. So the owner of a particular resource will have the ability to grant other users either ownership of that resource or read-only access or write-only access, and actually everything in between. So as Liz mentioned earlier, the most basic 3 types of roles that Terra provides are owner, writer, and reader, and those are probably fairly self-explanatory. But just to give you a very brief example, there's actually a lot of granular control that we can grant users with very specific use cases. So we can have different levels of access to public workspaces in which people can launch batch computations or are able to launch certain notebooks. We can give very specific permissions for people to be able to share certain access with others, and so on. So just to give you an idea that if you have a very specific type of use case in mind for example, a publicly facing workspace, that we have a lot of granular control over the types of access that you can grant. To that end, we also have a feature called authorization domain. You may have noticed that as Liz was taking you through cloning the workspace. At the very bottom of the pop-up to clone the workspace, there was a box for authorization domain that, as we said, we don't need for this particular demonstration. This is just an added security feature. If you're interested in it, authorization domains work essentially like laboratory badges. So it enables the person in control of the authorization domain to make certain that even if a user sends a share link to another user outside of the authorization domain, that the user who is outside of the authorization domain won't actually be able to use that workspace. Billing in Terra runs in the exact same way as the Google Cloud Platform. All Terra costs are billed via a Google billing account. So you actually have to set up a Google billing account and tether it to a Terra billing project. And just like with Google Cloud Platform, you're charged for storage, computation, and data egress. And so we have some instructions for people on how to both set up their own Google billing account and how to set up a Terra billing project to associate with it. And you need a billing project in order to clone a workspace and run a cloned workspace. So in the case of this demonstration, we're hoping that you're all signed up for the free credits project, which is a Terra billing project. And so you can use that to run a cloned workspace. Without a billing project, you can explore a lot of the public offerings, but you won't be able to launch any type of computation or perform any type of data egress, as those are the things that are billed. Billing projects, as I mentioned, are a type of resource, and there are a few different possible roles that a user can have on a Terra billing project. The owner is the person who initially creates the billing project and tethers it to a Google billing account, and they have full access, most importantly, including adding and removing other users to different roles. In addition to the owner role, there is a workspace creator role, which is very similar to the owner role, except that it does not allow the workspace creator to grant new access to other users. It only allows them to create additional resources on that billing project. And then notebook users and batch compute users are kind of like low-level users who should only be given access to the resources that are already in place. So how do we gain access to a Terra billing project? There are three ways. One is that the owner of an existing Terra billing project has to authorize you so that you can become an owner of that billing project. Another way is to set up a Google billing account and link it to a new Terra billing project. And simplest of all is clicking on the free credits program and activating it. So that automatically generates a billing project. And in case the name of that billing project looks confusing, they're automatically generated billing project names. So they start with the letters FCC and then have some random words. So if you see that in the list of your billing projects, that's your free credit account. So how do we set up a Google billing account? There's also a couple of different ways to go about that. First of all, you can create your own billing account using a credit card or bank account, and we have instructions for this in our knowledge base, and you can also find that information on Google's website. You can also talk to your own institutional procurement office to see if they have a preferred method for working with Google billing accounts. And finally, you can set up a Google billing account through a third-party reseller. And this is actually how our free credit program works. We have it set up through a third-party reseller called Onyx Networking. So the free credit trial, just so everyone knows, we'll just reiterate this. It begins as soon as you click the Start Trial button. That's when it activates. At that point, you will have the use of up to $300 on whatever it is you need, whether it be egress or storage or computation. This $300 will stay with you for 60 days, and you'll be notified by email once you have used 50%, 75%, and 90% of your credits to ensure that you have the time to migrate any type of work that you've done on your free credit account over to a normal account. However, just so everybody knows, if it so happens that you were working very hard on your free credit account and you generated a bunch of data that you would really like to keep, and it just slipped your mind that your 60 days were about to be up, and all of a sudden you find that you aren't able to download the data that you've generated, you can just contact us and we will gladly help you with that. Cost management is obviously very important for all of our users. To that end, we have actually quite a few options for keeping track of your costs. As I pointed out, the Notebook Runtime Environment button gives a constant real-time cost estimate for how much you're spending specifically on the Jupyter Notebook Runtime environment. You can also look for cost estimates within dashboard descriptions. So ideally, a dashboard description will contain a cost estimate for a given pipeline that tells you how long it should take to run that pipeline and how much money it will take to run that pipeline on a given size of input data. You can also use the Job Manager tab. You can also use the Job Manager, which is found through the Job History tab, to keep track of what costs you have accrued so far on your batch computations. And in addition to this, and I'll show everyone where to find this when we go back to the web application, we have a few really thorough articles on controlling cloud costs in our support knowledge base. So I'll show everyone where they can find those. So with that, I'll pause for about 30 seconds to see if anybody has any questions on anything we've talked about so far, and then we will go ahead and get back into the web interface. And I'll show you where you can find some of that documentation, and we'll get back to our hands-on exercise. So in the meantime, we'll go ahead and go back to the web interface. And you can see that our notebook runtime is operational. You can tell because our play button has turned into a pause button. So if we were to press this, this would pause our notebook runtime. We're not going to do that right now because we don't want to have to wait 2 minutes for it to shut all the way down and then another 2 minutes to spin all the way back up. In the meantime, we can start running computations in our Jupyter Notebooks. So I'll start in this notebook, the number 2 notebook, our environment setup. And I'll go into the edit mode. Hopefully everyone is following along. So here we are inside of our Jupyter Notebook environment. I'll point out that over here is where you can tell what sort of language the kernel is running at that moment. We actually support both R and Python. So if you wanted to change your notebook from being an R notebook to a Python notebook, you would go to the kernel menu and select Python as the kernel. you'll notice that this indicator indicates whether the kernel is currently working or if it's currently idle. So when the circle is empty, that means that nothing's going on and the kernel is just happily sitting there ready to do some work. If I force it to do some work, the dot will become filled in. So there you can see the kernel was busy for a little bit, and then it finished reconfiguring itself, and now it's idle again. Since this is an R notebook, I'm just going to go back to the R kernel. So here we are ready to run. I'm going to assume that everyone here is at least somewhat familiar with Jupyter Notebooks, so we won't go into too much of a nitty-gritty, but just so everyone knows, our Jupyter Notebooks support two different types of cells, and you can tell what type of cell you're looking at by selecting that cell and looking at this drop-down menu. And there you can see whether this is a code cell or a markdown cell. If it is a markdown cell, you have to double-click it to get into the edit mode. And then when you run it, it parses that markdown script and turns it into the nice formatted markdown cell that it should be. Another very popular way to run a cell is the shift return hotkey, which I will be using especially in the next notebook. In this notebook, rather than running through each one of these cells individually like that, I'm actually going to go ahead and press this button up here, which says restart the kernel and then rerun the whole notebook. So it's kind of a fast way of running an entire notebook all at once. And the reason I'm going to do that is because this particular notebook is just an environment setup notebook. So once all of these dependencies have been installed on this virtual machine, those dependencies will be ready in any notebook that you launch from this workspace. You'll also notice on the left hand here the active table of contents. It's a very convenient feature that we have enabled on all of our notebooks. You may have noticed that when a particular code cell is running, the line for that section in the active table of contents turns red, and once it finishes running, it turns white again. So it's a nice way of keeping track of how long it takes to run through an entire notebook. These are also clickable links, so I can click on one of these and it'll go directly to that section. So it's very convenient. So with that, we'll exit out of this notebook. And again, with the understanding that all of the dependencies that we've installed using this our environment setup notebook are now installed on our virtual machine, which is to say on this notebook runtime. And so now all subsequent notebooks will have those dependencies ready to go until you delete the notebook runtime environment. And incidentally, it is a good idea to do this about once every month or two to make sure that you are running the most up-to-date version of our available runtime environments, as they also tend to change. We do a lot of constant updating. So it is good every once in a while to delete your notebook runtime cluster and just press play again, just to recreate it. You don't have to do anything else other than delete it and recreate it. Unless, of course, you have dependencies, in which case you should run those dependencies again after you've recreated your cluster. So with that, I'm going to show you a little bit more around the interface itself. I guess I'll start by pointing again to our support pages. I wanted to make sure everybody knew how to find the controlling cloud costs documentation. We have quite a lot of documentation available in our support page. In particular, we have these two sections that I want to draw your attention to. The Getting Started section is for new users to get everybody up to speed in using Terra. There's actually also a section here for getting FireCloud users transferred onto Terra, in case you're already familiar with FireCloud. And then we have a lot more detailed documentation in this documentation section. For example, We have an entire section on controlling costs with a few articles in there. So if controlling costs is important to you, and of course it's important to everybody, this is where you can find that information. And this is also where you should go to read about any updates that are coming out in our blog section. This is also where you can go to request a feature in case you have noticed that there's some sort of very simple rudimentary action that you wish would be easier to do with a single button because you find yourself copy pasting a bunch of things from one place to another over and over. It's very likely that if the thing that you're doing can be done with the click of a single button, then we can create that button for you. So feel free to request a feature. Feel free to post on our community forum, which we moderate ourselves. And of course, feel free to contact us directly. So for the purpose of this exercise, we're actually going to go into the data library. So if you'll click on the hamburger menu, click on the library tab, and go specifically to the data section, we are going to construct our own custom cohort out of this 1,000 genomes low coverage data set. We're going to send that custom cohort to the workspace where we were just working, and then we're going to open our second notebook, and we're going to use that notebook to actually look at the cohort that we are about to create. So here we are inside of the Data Explorer for this data set. Every one of those data sets has its own unique Data Explorer. These are basically just graphic user interfaces that correspond to whatever the needs of those data custodians are. And it's a very convenient point-and-click interface. Right now, we could save this entire cohort. You can see technically we have all 3,500 participants saved, or rather selected, and we could save them by clicking save. If I were to click save right now, it would take me to a window that prompts me to select a workspace to send this cohort to. We're not going to select all 3,500 participants. We're going to be a little bit more specific. So it's very simple how you can pick different subsets. So for example, if I was only interested in samples that had this particular type of data, I would click on that. And as you can see, the number of participants in my cohort went down from 3,500 to 2,500. Just click it again to get all of those back. And for the purposes of this exercise right now, I'm going to select 3 populations. I'm going to go to the bottom of the population section, and I'm going to select people of Mexican. Oh no, pardon me. I went backwards by accident. We're going to select people of Mexican ancestry, Finnish ancestry, and Japanese ancestry. And so now we have exactly 317 participants. They belong to various genders and various superpopulations. The reason that I chose population is because the formatting of this field is very simple. You'll notice that some of these other fields have more symbols in the titles of the fields, and that makes it a little bit more tricky to query them in the notebook, although not impossible. So just for the sake of simplicity, we're going to select this field instead. And we're going to save this cohort. We're going to give it any old name. I guess we can choose the auto-generated name. Why not? Oh, cohort name has to only, so no spaces. So there we go. I'm going to save that. And it's going to ask me what is my destination workspace. So I know that this was called Webinar LK. So that's the one that we were working in just now. I'm going to click Import. And you'll notice that this cohort now appears in my data tab. Before I had done this, the only things that were in the data tab were these tables, the BigQuery tables and these two sample tables, which are a part of our batch compute demonstration. So here we have this cohort. Now we're going to go into our number 3 notebook, which is titled number 3, Retrieve a Cohort Using BigQuery. We're going to show you how you can actually look at this cohort directly. So I'm just going to zoom past the first section. The first section, for those of you interested in reproducing this exercise, the first section is just the instructions that I just took you through. It's the instructions for going to the data tab and generating your own custom cohort. So select filters for data, which is exactly what we did. Save the cohort to a Terra workspace, just as we did. And this, by the way, is a nice demonstration of the usefulness of Markdown as a language. So Markdown has a lot of convenient formatting for things like bullet points, but also for links and images. So we have a few other dependencies that we should install. And at this point, I'm going to start clicking through with the Shift Return hotkey. So this is how to run through notebook cells quickly, but one by one. So we need to do some of these preliminary steps. We need to make sure all of our dependencies are in place. We need to make sure that everything is up to date. We need to set our project global variables in order to make sure that our notebook understands that it's querying the cohort inside of this workspace. Incidentally, a lot of times you will notice that a cell will run successfully, even though it gives you this kind of big, scary pink warning message. Most likely, a warning message is not only not a bad thing, but a good thing. A warning message usually tells you that the cell that you run has completed as expected, but it might give you certain warnings, like, for example, that certain functions might be deprecated, just in case that affects your downstream analysis. You would expect a much shorter error warning without a color-coded window if something actually went wrong. And hopefully that won't happen in this demonstration. And so now we're going to load the cohort query. We're going to do a little bit of a sanity check. So the whole point here was just to make sure that we didn't get one of those errors. And again here, if we run the cohort query like so, then we actually can see all of the information about the query. So we can see the name of the cohort that we created, the entity type, which is of course cohort. We can see the data set from which this came, and a bunch of other information about the BigQuery query itself. So once we've done that successfully, we can start to view it directly in our notebook. So here I execute a cell that stores the BigQuery table inside of this variable, and we're going to examine the output. So here we are, and you'll notice that our table has 317 rows, exactly as we would expect. So this was the custom cohort that we created. You'll notice that this table currently only has one column, which contains the participant IDs. So you can see up here, 317 by 1 means we have 317 rows and one column. We want to look at some more data in that cohort. And so to that end, we're going to have to join additional data into that column. We're going to do that using these couple of cells. You'll notice that the data that I'm going to be joining in is the gender field. So you can see when we print out the dimensions of our newly merged table, we now have 317 rows as before, but now we have 2 columns because we've appended the gender column to our table. And now we're going to go ahead and plot that so that we can see exactly the breakdown of gender in our cohort. So we have 172 female participants, 145 male participants. We've printed out both the numerical representation and this nice plot. And finally, of course, we have a field to produce the provenance. So that gives you the information for all of the tool versions that were used in generating this notebook. So as I mentioned earlier, I did want to show you how to change this from a gender to a different field. So the field that I selected for was population. So I'm just going to change this gender to population over here, and I'm going to make sure to do that everywhere else where I need to. I believe that would be these 2 places. And we'll go ahead and rerun everything from here down. And there you can see our 3 populations: Finnish, Japanese, and Mexican ancestry. So it's that simple. It's very nice. It's a very good-looking way to generate outputs. And that brings us to the end of the demonstration of notebooks. Again, feel free to post any questions that you have in the chat as we go through this. The last portion of the demonstration will be to launch a couple of batch computations. So to that end, we're going to switch over to our Workflows tab. And we'll start by doing the simpler exercise, which is titled Cram to BAM Exercise 1. This one is completely pre-configured for you. So it's really this simple. All you have to do is click Run Analysis. However, I'll just show you around a little bit. When you're in the Workflows tab and you click on a particular workflow, you can see, first of all, what version of WDL is being used in that particular workflow. You can select exactly which data you want to run on. So for example, if you have a workspace that has a very, very large data table and you don't necessarily want to launch a batch compute job on the entire data table, you can choose specific rows. We're not going to do that in this case. We're just going to go ahead and stick with our pre-selected samples. And we're going to click Run Analysis. And this takes us directly to the Job History tab, which shows us that our 2 batch compute jobs are currently queued. Queued should turn fairly quickly into submitted, maybe not quite that quickly, but then it will stay submitted until it either returns as having failed or having succeeded. And you'll notice if you go to the Data tab, we've generated a new data table. So this is the output of the job that we just launched. So with that, I'll just show you the ever so slightly more involved example. So in the Cram to BAM exercise 2 example, we do the exact same thing. However, you'll notice that the Run Analysis button is grayed out. And the reason is because at least one required attribute is missing or invalid. So let me tell you a little bit about what you can see in this portion of the interface. In the inputs, you can see what the inputs are. You can see the task name and the variable name, as well as the type of object for each one of those, as well as what are called attributes. which in many cases are actually the file names. And so in the previous exercise, all of these were filled in. However, in this exercise, we actually need to click here, and there are some auto suggestions. And we have to choose the correct options in order to be able to launch the workflow. And you can tell which fields have to be filled in based on this icon. See, it says this attribute is required. The same thing can be true in outputs, where you can fill in the attributes yourself. And in the script tab, you can actually look at the raw whittle script for that particular workflow. You can't edit it in this view, but this gives you an easy way of looking at the whittle script and deciding if you need to modify it somehow and re-upload a different workflow. So in this example, we'll just go ahead and choose correct options. I guess since we don't have very much interaction, I'll just go ahead and choose them. So in this case, we want our ref index. We want to find that. There it is down there. Here we want to find our ref FASTA, like that. And we want to make sure to point our workspace in the correct direction by selecting an attribute for the input cram. And then we have to click save, and now we can run our analysis again. Oh no, there was a problem. What could that have been caused by? Perhaps this is not the correct sample ID. And so this is actually a part of the exercise that we wanted to leave to the users to try out on their own. And we do have specific instructions on how to do this in the dashboard. So everyone can go ahead and enjoy playing around themselves. Just so everybody knows, there's in the job history tab a very convenient way to relaunch jobs in case they come back with a failed status. It's very simple. And that really takes us through all of our hands-on exercises for this webinar. So with that, I'll be happy to take any additional questions you might have. Again, here's an overview of what we've done so far.
We have an outstanding question about if they need to edit a script. What do they do if they want to edit a script? If
you want to edit the whittle script?
Yes.
So if you want to edit a whittle script, so you can't unfortunately edit a whittle script directly. There you go. Yeah, so this is unfortunately not an editable field. This is a read-only field. If you want to edit a whittle script, you would copy this whittle script into whatever environment you like to do whittle work in, create whatever edit you like, and then you will have to upload that workflow, which happens in actually a different portion of the interface. Then you would find that workflow using this button, and then it would populate your newly edited workflow into this section.